In the next chapter, we'll look generally at hidden messages, in particular in genomic sequences. And there will be some cute and maybe surprising and maybe insightful math about this. But before we get there, I want to set a bit more of the context. And so in a way, this is what in this module maybe should have been in week 1, but I wanted to start with some computer sciencey fun. So let's catch up a bit on the background from biology. I said I'm not assuming any biology knowledge for this module, so I wanted to stay true to that and equip you with the little things that might be relevant or necessary to make sense of the puzzles we're discussing, which isn't going to be a lot. But I hope to pique your interest so that you dig a bit more into this, just because it's interesting to know, and there's so many exciting things to learn in science. Biology is the study of living things. I'm not going to get into the definition of what living means in this case. It's surprisingly controversial. I think the usual is it has some metabolism, it generates energy, it has some way of reproducing, and it has some way of compartmentalization. There's some sort of stability over time. You don't just disintegrate into space. I'm not sure if that's universally agreed, especially for viruses. This is excluding them from living things because they don't have a metabolism. Okay, a little bit of the kind of evolution, the history of the field, maybe better to say. I think biology had, for a long time, essentially consisted of people that were excited about nature, observing what happens in nature and writing these things down and drawing beautiful pictures. And of course, doing a lot of structuring life in the way they could see it. Maybe I'm not doing them justice by saying that they didn't do controlled experiments to the extent that biologists would do now. An example, if you want to read up about this, are the Darwin's finches. The beaks of these birds show high variability, and that's reported to be an eureka moment for Darwin in his theory of evolution. That he said these beaks evolved to solve specific functions. Okay, maybe this is a bit unfair, gradually more scientific, but people tended to use the scientific method more systematically. A nice example with a German background is Mendel's inheritance theory. So there's experiments with certain peas, and he specifically tried to check how are certain traits inherited. And then more and more things moved from what you can see with your naked eye to smaller and smaller things. It turns out life is complicated. And if you're trying to understand the mechanisms at the macro scale or at our scale, it's essentially imperative to understand the lower levels first. A big thing for biology, therefore, was the development of microscopes, first optical and then more refined versions. And I guess it's now quite clear that the origin of life was microscopic, and also that many of the fundamental mechanisms can only be understood at this microscale first, and then you can try to scale up with all the complications that come from the complex systems that living things are. Okay, we'll try to dig a bit into this. The main thing that happened in the 20th century, from my humble perspective maybe, is that things moved much closer to computer science. It was the discovery of DNA and genes, and in particular that things are much more discrete than maybe they first look. I'll show this video a bit because it nicely shows a bit of the structure of DNA that I can't possibly explain in the same visual way. But from a computer science perspective, you could say your genetic information stored in DNA is one long string over these 4 bases. Pretty much nailed down to this 3 billion letters. As always in biology, nothing's completely exactly written in stone and there's variations. But the rough number is clear, so you can work out how many bits of information that is. The somewhat depressing fact is, I don't know, some 4 gigabytes, roughly. The more depressing fact is if you take the difference between two of us in the room and take your genetic information as the encoding of your difference from your neighbor, that's roughly 30 megabytes, so you could email it. Okay. Because the information is discrete, it's one of those 4 bases and not too much else, you can discretely copy it and you can make precise copies. That's what we love about digital data. And turns out life invented that much earlier than we did. And then mutations are what happens if there's mistakes in the copying, among other things, as always. Okay, for the structure of, I'll come back to to this function, but let's first have a look at what the structure looks like.
Large organisms like humans are made up of organ systems, such as the circulatory system and digestive system. These systems include a number of organs. One of them is the liver.
We'll get there in a moment. They want to show off their 3D
and these
modeling
organs are
skills
made up
first.
of tissues. And these tissues are built of cells, each specialized to perform a particular function. Within each cell are structures and compartments called organelles. These include the cell membrane, mitochondria,
You don't have to remember the names, right? It's,
The nucleus contains 23 pairs of chromosomes, including a pair that determine your biological sex. Each chromosome is one long length of tightly coiled DNA. Unwind it, and we see the DNA is wrapped around proteins called histones. The DNA is a double helix, like a twisted ladder. It is made up of 4 nucleotides, which contain different bases: adenine, cytosine, guanine, and thymine. A, C, G, and T. A always pairs with T, and C always pairs with G. The order or sequence of these bases in specific sections of the DNA, called genes, carries the instructions for a cell's structure and function. One copy of the entire DNA instructions for an organism is known as the genome. The genome, organized in chromosomes within the nucleus, tells each specialized cell how to function with its neighbors. These make the tissues that together build organs, which work in organ systems and collectively enable an organism to grow and live.
yeah, it's made by Your Genome is a British not-for-profit that makes some. They're funded by, or they're working with the Sanger Institute on bioinformatics research. Some of named after one of the first people that sequenced DNA, and they have this as part of their outreach program. I think the visuals are compelling. It's much nicer than I could possibly draw it. It's also somewhat scientifically accurate to my knowledge. So you see there's more to it than just the abstraction of one long string. On the other hand, it is really one long string to a good degree of approximation. In particular for working with it and the function of it, that's often what we can do. One thing we're relying is certainly that the chromosomes are not really connected. It's not one long string, it's one string per chromosome at best. That's also if there's websites where you can look at the human reference genome, for example, and zoom down to all the A, C, G, and T's. Not that these by themselves help us often that much, but there you usually have to first select which chromosome you're looking at. Okay, so the main amazing part for me is that there's so much computational components in there. There's copying, there's data, there's more coming in terms of translation, and essentially little computer-like processes going on. It's certainly a distributed and parallel processor, different from the centralized way of thinking that we prefer, but it has computational components, which I think is mind-blowing enough. But it also means computerized methods can help a lot in advancing biological understanding. Now, the latest twist to that, of course, data science and AI comes also into this, because all of the computational things aside, there's enough chaos and complication in the systems that more fuzzy techniques can often help. But I'll focus more on the parts of bioinformatics where there's a clear edge for discrete and combinatorial algorithms. Last exciting thing is that sometimes you can replace biological experiments with in silico experiments, so you simulate. And that's a mainstream part of biology research these days. Okay. We won't make it through the second video I want to watch with you. We'll do that next time. But I want to show you what I started preparing, namely it's both a collection of resources that I like, but also a tool. You didn't see the slides. So there was some text accompanying what I said, but not much beyond what I told you verbally. Right, let me show this briefly. So I prepared a little mind map with just various links, and it's my attempt to involve you a bit in this. So it's a collaborative mind map. The link is on ELS and CampusWire if you want to join. I think some people already played a bit with it. So just very briefly, it's a cool tool in itself. It's a German startup, Infinity Maps. The basic thing is a nested mind map. So you can always zoom in, and you can zoom in indefinitely, and you can have a lot of complexity baked into some specific part, but then immediately zoom out and say, oh, what's the big picture again? Here we are. So you can try to use it to organize knowledge, whether you want it or not. You can do that on your separate account. You can try it for free. What I tried is to structure a bit the resources I know, and which I think are good, especially because I have trust in their scientific accuracy to some degree, but they're at various levels of being whimsical and fun and internet stuff. There's the videos from the book that I mentioned, Bioinformatics Algorithms. There's also some great videos from Ben Langmead. He's a bioinformatics researcher, produced one of the main tools for read alignment. The tree of life is this idea that you can cluster all the species according to where they diverged in biology, in evolution. I encourage you to look at that as well. And there's a few more YouTube channels, some of which we'll see videos from. Yeah, so have a look at that and add cool things that you find. If you're not sure whether they're just fun or also scientifically useful, maybe just add a comment of that sort. Up to you to make something out of this.